In Chapter 3, we're going to look at cells. So first of all, we need to always start with some basic definition. What is a cell? A cell is defined as the smallest unit of life, and we'll look at some of the basic components you need to have to be a cell throughout this chapter. Cells were first observed by Robert Hooke in the 1600s. He kind of ground fine lenses down and was able to look at plant material and different things, and he described them as being kind of irregular box-shaped structures. It reminded him of the prison system at the time, so he called them cells. He was actually looking at a section of a cork tree. So that's where we get the name or the term cells. Since then, cells have been studied quite a bit more, and we now have the cell theory. And this was a combination of several different scientists that looked at basically everything they could get their hands on, plant material, animal material, and found that all living things are made up of one or more cells, and also that cells only arise from other pre-existing cells, so they don't spontaneously generate. So for every cell that exists, it came from a previous cell that divided. And pretty much everything is made up of cells, so fungus, plants, this little head louse, which is gross to look at. Different examples of cells would be a hummingbird egg and an elephant egg. Those are different size cells. We've got beluga sturgeon eggs, and also human eggs. And an interesting thing about human eggs is they are the most expensive eggs. So ladies, if you want to donate your eggs, you can have hormone injections, a very invasive procedure, and donate your eggs to women who simply can't produce their own eggs. Why are cells so small? It has to do with surface to volume ratio. So the smaller a cell is, the more volume or surface it has on the outside compared to the volume inside. As it gets bigger, that ratio changes, and it basically becomes more difficult for the cell to get rid of waste products and also to have enough surface area to bring in nutrition and other things into the cell. So cells remain small because of that surface to volume ratio problem. To look at cells, what we're going to use in class are light microscopes, and we have compound light microscopes because they have two eyepieces. And ours are fairly good microscopes that we have here in class, but they have the same limitations that all light microscopes have, as they use a wavelength of light. And because light travels in waves, anything that's less than half of a wavelength, so if we're looking at a virus that's less than half of a wavelength, we simply cannot see it in a light microscope. It's invisible. So to see things smaller than half a wavelength of light, we need to use an electron microscopes. And these, instead of passing light through your sample, these actually, it's done in a vacuum chamber, and they shoot electrons through your sample, and then they're picked up by detectors and read. And there's different types of electron microscopes. There's transmission electron microscopes, where the electrons actually pass through your sample. And I should point out, these two pictures here are different types of light microscopy. This one shows transmission electron microscopy, and you can see very fine detail of the different structures within these cells. There's also scanning electron microscopy, and for that you coat your sample with a very thin layer of a heavy metal. Gold is commonly used. That's what I always used when I did electron microscopy. And you coat your sample with a very thin layer of this heavy metal like gold, and then you shoot electrons at it, and they bounce off, and they're read by detectors, and you get very, very detailed three-dimensional images of the outside of your structure. And that's very cool because you can get some really neat pictures of things that you may not want to look so closely at. For example, these adorable little critters. These can sometimes be found on humans, and we can see it's got some nice little antennae. By the way, this image down here is what it normally would look like while you're using the electron microscopes, and all of these are just false color images, so they've added color to them so that you can see the structure. So we've got nice red claws on this one. This one over here is actually grasping a hair shaft. This one is very cute because you can see its little face. Right here is its mouth, and it actually feeds using a proboscis, so a long skinny tube like a butterfly. These are less friendly than butterflies. These are actually crabs or pubic lice. So aren't you glad we have electron microscopies to get a really good look at these guys and see how friendly and cute they are? These are very common around college campuses, but please in class don't share any stories that you might have of these. Looking at the different types of cells, the first type of cell is a prokaryotic cell, and they're fairly basic in their structure. They have a plasma membrane, and we'll talk more about that membrane in the next section of the chapter, but the plasma membrane would be kind of in here, and it holds the whole cell together. It's got the cytoplasm, and that's the fluid that fills the cell, and that fluid is going to be concentrated with nutrients and things the cell needs to survive. It has ribosomes, and the ribosomes are these little purple specks that are all over, floating around in the cell, and the ribosomes make protein inside the cell. It's got DNA, two different types of DNA in our prokaryotic cell. We have one main chromosome, or this big loop here of DNA, and that's its main chromosome. It also has smaller loops of DNA called plasmids. And we'll get a chance to look at those plasmids, and in fact later on in the semester we will actually transfer a plasmid, so a small circle of DNA like this that contains information for making a glowing protein, and we'll transfer that plasmid into E. coli, and we'll genetically engineer those E. coli to fluoresce under UV light, which is very cool. So we'll work with bacterial DNA later on in the semester. There's also a cell wall, and the cell wall is this thick brown layer on the outside, and the cell wall is very important for bacteria because it holds them together and gives them their shape. Because bacteria can't really control their environment, any nutrition or anything like that that they encounter in their environment, they will immediately start packing it to the inside of their cell as fast as they can, and what that does is it creates pressure. So the average bacterial cell is under about the same amount of pressure as the tires on your car. And many antibiotics work simply by clipping away at that outer wall. Once it's weakened, the bacteria just pops open because it can't hold itself together anymore. Some bacteria also have flagella or other ways to move, and this is our flagella coming out of our cartoon bacterium. Here's an actual picture of a bacteria. It has 3 flagella coming out of it. Again, not all bacteria have the flagella to move, only some of them do. The second type of cell are eukaryotic cells, and I should also mention prokaryotic literally means before, so pro is before, and karyote is the nut or kernel. So notice there's no big blob inside here. So when scientists first started looking at cells through the microscope, the first things that they noticed is these prokaryotic cells didn't have any big structure inside. However, the eukaryotic cells, this is after the nut or kernel, do have this very large structure that's easily seen even in our light microscope. So we'll be able to see this in a week or so when we do our cell division lab. We'll see the nucleus inside cells. And that's the defining characteristic between prokaryotic and eukaryotic cells, is eukaryotic cells have not only the nucleus, but also lots of other compartments inside that all have specialized functions. And all those compartments are surrounded by a membrane, which we'll talk more about in this chapter. So this figure just compares our prokaryotic and our eukaryotic cells, and there's lots of different types of eukaryotic organisms. In fact, you are a eukaryotic organism because all of your cells contain a nucleus and organelles. There are different types of eukaryotic cells. There's animal cells and plant cells, and they have some main differences. And by the way, one of my favorite questions to ask on the exam is to show you a picture of either a plant cell or an animal cell and ask you which is it? Is it plant or animal, and why? So we'll look at some of those different internal compartments and structural elements. So by the end of this chapter, you'll be able to easily recognize that this one over here is an animal cell, and that this one over here would be a plant cell, and name the structures that make them that way. Since we've already looked at kind of the cell theory and that all cells arise from other pre-existing cells, we want to look at kind of how the first types of eukaryotic cells were produced. And they came about through simpler types of cells that didn't have any membrane-bound compartments. And there were two processes at work to generate our current eukaryotic cells. The first one was probably something called invagination, where the outer edges of the cell started folding in to make kind of little cavities and ripples. And this does a couple of things for the cell. It will help trap food and other nutrients that are on the outside and give it time to digest it and absorb it. It also increases the surface area, since one of the things we've talked about already that cells have a problem with is surface area. In fact, invagination is used a lot to increase surface area. Your entire digestive tract is all invaginated to increase that surface area to absorb your food and nutrition that you take into your body. So endosymbiosis is the second type of process that probably occurred for cells to go from prokaryotic to eukaryotic. And an example of an organelle we got through endosymbiosis would be our mitochondria. So mitochondria, through a variety of techniques looking at the DNA and the structure and how they function, they actually were a separate living species that was engulfed or kind of eaten by a larger prokaryotic cell. And then because they were mutually beneficial to each other, that's what symbiosis means, that they're mutually beneficial, these two cells kind of lived together. This one producing food energy for the larger one to use, and the larger one just giving the smaller one a good environment to survive in. Over time, they began reproducing together, and the smaller one lost some of its characteristics that would allow it to live or exist outside of its host cell. So now they're just kind of permanently together, so they can't survive one without the other. So those two processes helped give us our eukaryotic cells. And plants also have chloroplasts, which are the organelles that capture light energy from the sun, and they came in by the same process as mitochondria did.